Hello everyone. How did Griffith unveil the transforming principle? Do you want to know? So let's go into the topic. Frederick Griffith was a British bacteriologist. His experiments with bacteria were the first to reveal the transforming principle, leading to the discovery that DNA acts as a carrier of genetic information.
In 1928, Griffith did a series of experiments with Streptococcus pneumoniae, a bacterial strain responsible for causing pneumonia. So he witnessed a miraculous transformation in this bacterial species during his course of experiments. So during the course of experiment, he found that the bacterium has two strains basically, the virulent type, the S type strain, forming smooth colonies as it has polysaccharide sheath covering it, and the non-virulent type, the R type, forming rough and irregular colonies. They do not cause any disease. Now let us take the look on Griffith experiments. So he did a series of experiments with two types of bacterial strains, the virulent type, the S type, which is causing disease, that is pneumonia, and the non-virulent type, the R type, which is not going to cause any disease. So first, he injected the S type strain, that is the virulent type, into the mice. As we know, this S type is the virulent type. It caused disease, that is pneumonia, and the mice was found dead. And next, he proceeded with the R strain, that is the R type, which is supposed to be the non-virulent type. So now it was injected into the mice, and the mice, it was alive. Of course, we know because the R type is a non-virulent type, so it didn't cause any disease. As a result, the mouse was alive. So right now, he did the third one with smooth strain, that is the virulent type, but it was heat killed. So he injected the heat killed bacterial cultures, that is the S type cultures, the virulent type cultures, into the mice. Now the mice was alive because the cultures were heat killed. So it didn't exhibit any virulence in this species. So the mice was found alive. Now the fourth set of experiments. In this, he used the combination of bacterial cultures. He used both the S type, which was heat killed, which was a virulent one, along with the live non-virulent strain, which is the R type. So as you can see in the picture, here the S type cultures were heat killed, and the R type cultures, it is live. So he used the combination of cultures here. It was injected into the mice, but here the mice was found dead. So it was concluded through this experiment that some factor in this heat-killed virulent strain, that is the S strain, it could have transformed this R type, that is the non-virulent bacterial strain, into the virulent strain, so which must be due to the transfer of genetic material. So the non-virulent R strain has gained virulence via some factors which has been transformed from the S strain to the R strain due course. So this is how the transforming principle has been unveiled.